The global health supplement market is worth approximately $1.5 trillion. Every month, millions of people spend significant sums on NMN. Resveratrol, coenzyme Q10, collagen peptides, and glutathione molecules packaged in attractive bottles. With promises of cellular rejuvenation and reversed aging, science tells a different story. The most powerful cellular repair mechanism available to the human body is not sitting on a pharmacy shelf. It is built into the silence that follows your last meal of the day. Today we are going to walk through 8 molecular mechanisms by which fasting heals cells more completely than any supplement currently on the market. Before the mechanisms, the evolutionary logic needs to be clear. Humans evolved across millions of years of irregular food availability. The absence of food was never a cellular emergency. It was the scheduled signal for maintenance, repair, and recalibration. Cells operating in nutrient abundance dedicate energy to growth and division. Cells operating in nutrient scarcity switch energy allocation to repair and cleaning. The molecular switch governing this transition is the balance between mTOR and AMPK. When food arrives, mTOR activates and drives protein synthesis and cell proliferation. When food disappears, mTOR is suppressed. AMPK activates, and cellular repair, fat oxidation, and autophagy engage. The modern problem is that this switch is locked in the mTOR direction around the clock. Breakfast, lunch, dinner, snacks, late-night eating. Cells never receive the signal to enter repair mode. The first and central mechanism is autophagy, the cellular self-digestion process that earned the 2016 Nobel Prize in Physiology or Medicine. Autophagy is the process by which cells identify damaged protein aggregates, dysfunctional organelles, aged mitochondria, and pathogen remnants inside themselves, package them into autophagosomes, and break them down for recycling. This is not housekeeping in a metaphorical sense. It is the literal dismantling and rebuilding of cellular machinery, the process that prevents the protein waste accumulation, driving Alzheimer's disease, Parkinson's disease, cancer, and cardiovascular disease. Based on research published in PMC, autophagy begins activating progressively between 12 and 14 hours of fasting, with dramatic amplification in hepatic and neuronal autophagy responses at 24 to 48 hours. A study confirmed dramatic upregulation of neuronal autophagy during short-term fasting. Most significantly, a landmark 2024 paper published in Nature Cell Biology established that fasting and caloric restriction raise intracellular spermidine, a polyamine in yeast, flies, mice, and human volunteers simultaneously, and that this spermidine acts as an essential mediator without which fasting-induced autophagy cannot proceed. Fasting generates its own autophagy-triggering compound inside cells. The body manufactures it without any external supplementation. The second mechanism is the explosive release of growth hormone. In adults, growth hormone drives cellular repair, muscle recovery, lipolysis, skin collagen synthesis, and immune regulation. It is one of the most potent anti-aging hormones in the body. Research reported through EurekAlert documented that during 24-hour fasting periods, growth hormone rose an average of 1,300% in women and nearly 2,000% in men. This magnitude of growth hormone elevation occurs through pure physiological stimulation, no injection, no secretagogue supplement, no prescription required. At these levels, cellular repair accelerates, muscle protein synthesis is supported, collagen fiber remodeling in skin proceeds, and visceral fat lipolysis intensifies. The equivalent response cannot be achieved through any over-the-counter supplement. The third mechanism is NAD elevation and sirtuin activation. NAD is a coenzyme essential for cellular energy, metabolism, and DNA repair, whose concentrations fall by roughly half between age 20 and age 40. This decline is increasingly recognized as a primary driver of mitochondrial deterioration, impaired DNA repair, and accelerated aging. Caloric restriction and fasting raise the intracellular AMP to ATP ratio, activating AMPK, which promotes NAD biosynthesis. Elevated NAD activates sirtuin proteins, particularly SIRT1, and SIRT3 which execute DNA damage. Repair, regulate gene expression through histone deacetylation. Maintain mitochondrial health, and suppress inflammatory signaling. NMN and NR supplements are sold precisely to raise NAD levels and activate this pathway, at prices exceeding the equivalent of multiple months of groceries per bottle. Fasting activates the same AMPK NAD sirtuin cascade at zero cost, with the added advantage that the NAD produced is synthesized directly within the cell at 100%. Bioavailability: no intestinal absorption efficiency concerns. No molecular stability questions. The fourth mechanism is mTOR suppression and its anti-aging consequences. mTOR is the master regulator of cellular growth. In nutrient abundance, mTOR drives protein synthesis and cell division, but simultaneously suppresses autophagy and accelerates cellular senescence. When fasting suppresses mTOR, cells exit growth mode and enter repair and survival mode. Unnecessary protein synthesis ceases, damaged components are cleared, and the cell cycle is recalibrated. Rapamycin, a drug that pharmacologically inhibits mTOR, has attracted enormous attention in longevity research for its life-extending effects in animal models. A 12 to 16-hour fast produces meaningful mTOR suppression through natural physiological means, activating autophagy and cellular repair pathways simultaneously, without a prescription. The fifth mechanism is mitochondrial repair and biogenesis. Damaged mitochondria accumulating in cells increase reactive oxygen species production, generating the oxidative damage to DNA, proteins, and membranes that drives accelerated aging. Fasting addresses this through two complementary processes. Mitophagy: the selective autophagy of damaged mitochondria removes dysfunctional units. Mitochondrial biogenesis: the generation of new mitochondria replaces them. During fasting, AMPK and SIRT1 activate PGC1 alpha, which directly orchestrates mitochondrial DNA replication and new mitochondria assembly. A 2024 study published in Frontiers in Nutrition confirmed that intermittent fasting promotes neuroprotection through ketone body generation, enhances mitochondrial function, and reduces oxidative stress in a coordinated cascade. The ketone body beta-hydroxybutyrate acts not only as an energy substrate but as an antioxidant signaling molecule that directly protects mitochondria and suppresses reactive oxygen species production. The sixth mechanism is complete immune system regeneration. During extended fasting, circulating white blood cell counts decline, and this depletion triggers hematopoietic stem cells to initiate self-renewal and generate new immune cells during the refeeding period. Aged, damaged immune cells are eliminated and replaced with functionally healthy new ones. A structural reset, not a boost. No supplement category achieves this. Probiotics, vitamin C, zinc, and elderberry extracts all support the functioning of existing immune cells. Fasting replaces the immune system itself. The seventh mechanism is the structural reduction of chronic inflammation. Chronic low-grade inflammation, elevated CRP, IL-6, TNF-alpha is the shared molecular foundation of cardiovascular disease, type 2 diabetes, cancer, dementia, and depression. Fasting suppresses the nitrogen monofluoride kappa-B pathway and blocks the NLRP3 inflammasome through beta-hydroxybutyrate, reducing inflammatory cytokine production at its source. A meta-analysis published in Frontiers in Nutrition found that intermittent fasting with 6 to 10% weight loss reduced HSCRP by 18 to 48%. IL-6, TNF-alpha, and IL-1 beta all showed significant reductions after 2 or more months of intermittent fasting across multiple meta-analyses. No supplement achieves comparable systemic anti-inflammatory effect. Omega-3 fatty acids, curcumin, and resveratrol show anti-inflammatory properties in isolation, but their individual effect sizes consistently fall below the whole body. Inflammatory suppression produced by fasting. Safety must be addressed directly. People with type 1 diabetes, hypoglycemia risk, active eating disorders, serious underweight conditions, or those who are pregnant, breastfeeding, or under 18 should not independently implement any fasting protocol. Without explicit medical supervision, anyone taking blood glucose-lowering medications, anticoagulants, or corticosteroids must consult their physician before changing meal timing, as fasting alters drug metabolism and effect timing. Stop any fasting protocol immediately if severe dizziness, persistent headache, or cardiac palpitations occur. Cells do not wait for repair materials to arrive from outside. They wait for the supply to stop. Autophagy dismantles the wreckage. Growth hormone rebuilds the structure. NAD wakes the sirtuins. mTOR falls silent and repair mode activates. Mitochondria are renewed. Immune cells are replaced. Inflammation quiets. The brain clears. 8 layers of healing, all triggered by a single switch: the absence of food. Not one expensive capsule. The decision to put down the fork is the most evidence-supported, most biologically complete, and most cost-effective cellular repair intervention available to any human being alive today.